Welcome to my channel. In this video, I am going to solve numerical problems that appeared in IIT JAM Biotechnology examination. In the year 2018, two videos are already uploaded. In those, I have solved 2016 and 2017 paper. This video not only helps you to prepare your JAM exam but also for other exam too. So let's start. The first question. In this problem, glucose is incubated with glycolytic enzymes ATP that are radiolabeled with heavy isotope of phosphorus at the gamma position and unlabeled inorganic phosphates. At the end of glycolysis, which products will be radiolabeled? To solve this type of problem, you have to remember the glycolytic pathway, especially those steps in which phosphate group are used. In this reaction, ATP is used and product is glucose 6-phosphate. Here ATP is radiolabeled, so glucose 6-phosphate will be radiolabeled. In the same way, another ATP is used in this reaction and the product is fructose 1,6-bisphosphate. In this reaction, inorganic phosphate is used, which is unlabeled in this case. In 1,3-bisphosphoglycerate, 1-phosphate will be unlabeled cause it comes from unlabeled inorganic phosphate. So option C is incorrect. Now the last option, it is also correct because it comes from ATP which is radiolabeled. So option A, B and D are correct. In this problem, you have to calculate which of the following ratio is or are equal to 1. In double-stranded DNA, we know that according to Chargaff rule, in double-stranded DNA, adenosine plus guanosine is equals to cytosine plus thymine, and adenosine equals to thymine and guanosine equals to cytosine. Or we can say that adenosine and thymine ratio is 1, guanosine and cytosine ratio is 1, and adenosine plus guanosine and cytosine plus thymine ratio is always 1, and guanine plus thymine and adenosine plus cytosine is also 1. Here option B and D are correct. In this question, you are asked to find out the number of genetically different types of gametes will be produced by this heterozygous plant. The genotype of this plant is provided. In order to calculate how many type of gamete it will produce by this plant, the first you have to find out the number of heterozygous allele present in the given genotype. Here in the above genotype, capital B and small b and capital C and small c are the two heterozygous alleles. Capital A and capital A is homozygous allele. Now we will use 2 to the power n equation to calculate the number of gametes. And here n is equal to 2. So number of different types of gametes will be produced by this plant is 2 to the power 2 or 4. Now in this problem, two strands of the DNA are radiolabeled with radioactive thymidine and then allowed it to replicate in an environment containing non-radioactive thymidine. You have to calculate the number of radiolabeled DNA strands present after 3 cycles. In this experiment, DNA was first radiolabeled, that means both strands will contain radioactive thymidine. Now if DNA is allowed to replicate in non-radioactive thymidine, due to the semi-conservative nature of DNA replication, only newly synthesized daughter strand will be non-radioactive, but the initial 2 parental strands will be radiolabeled no matter how many times they are allowed to replicate. So the answer will be 2. Next, you have to calculate the number of beta oxidation cycles required to complete degradation of palmitic acid. In beta oxidation, we know that 2-carbon compound called acetyl-CoA is released from the fatty acid at the end of 1 cycle. If the number of carbon atom in the fatty acid is 16, at the end of the cycle it will be 16 minus 2 or 14 carbon compound. In this way, after 6 cycles, 6 into 2 or 12 carbon compound will be liberated from the palmitic acid and it will become 4 carbon compound. In the 7th cycle, the 4 carbon compound will break down into 2 carbon compound. So 7 cycles are required to completely break down the palmitic acid to produce 8 molecules of acetyl-CoA. Here is the second way to calculate the number of cycles required to completely break down a fatty acid. First, check the number of carbon atoms present in the fatty acid. Here in this case, it is 16. I have already discussed you that 2 carbon compounds will be liberated from fatty acid at the end of each cycle. So 16 by 2 or 8 2 carbon compounds will be liberated after complete breakdown of the fatty acid. So the number of cycle will be 8 minus 1 or 7, because in the last cycle 4 carbon compound is break down into 2 carbon compounds. In the next problem, the concentration of an enzyme solution is provided. 10 microliter was used and mixed with 1 ml reaction mixture. The enzyme can convert 20 nanomoles of product in 1 minute under optimal condition. Then you have to calculate the specific activity of the enzyme. Here we will first calculate the enzyme concentration in the reaction mixture. The concentration of purified enzyme is 10 mg per ml. From this solution, if 10 microliter enzyme solution is used and mixed in 1 ml reaction mixture, then the concentration will be 0.1 mg. Now we will calculate the specific activity using this equation, because we know that the amount of enzyme that catalyzes the reaction of 1 nanomole of substrate per minute under standard condition is 1 unit. So specific activity is nanomole of product form by minute by amount of enzyme in mg, or 20 into 0.1 or 0.2 unit per mg in this case. In this problem, a 100 nucleotide long single stranded poly A is synthesized from AMP. The atomic mass of carbon, hydrogen, oxygen and phosphorus are provided, and the molecular mass of AMP is also provided. Then you have to calculate the molecular mass. When one phosphodiester bond is formed between two AMP molecules, one molecule OH group is liberated. The molecular weight of OH is 1 plus 16 or 17. When 100 AMP molecules are bonded, 99 molecules OH group will be liberated. So 99 into 17 or 1683. Now the molecular weight of 100 AMP is this. So the molecular weight of 100 nucleotide long poly A tail will be the molecular weight of 100 AMP minus the molecular weight of 99 OH group. So it now comes to 32,817. In this problem, you have to predict the percentage of colorblind boy child will be born if the colorblind lady marries a normal vision man. When a colorblind woman marries a normal male, daughter will be carrier for the disease and all sons will be the colorblind or affected. Look at this table and you will realize that in this case of boy child, the X chromosome is always comes from the mother only, which carries colorblind alleles. So the correct answer is all sons will be colorblind or 100% affected. In the last problem, you have to calculate the pH at which it carries no net charge. We know that the pH at which the amino acid carry no net charge is called isoelectric point or pI, and it can be calculated using this equation. Here pKa is 3.1, pKb is 8 and pKr is 10.8. So pI is 9.4. So that's all for now. If you like this video, press the like button and share with your friends. And if you are watching my videos for the first time, press the subscribe button and press the bell icon. Wish you all the best for your upcoming examination. And finally, thanks for supporting me.